Today, we're going to have a look at the transport systems that are responsible for the transport of different organic materials in plants. Now, first, we're going to have a look at xylem vessels, which are responsible for the transport of water and minerals from the roots to the stems and the leaves. If we had to have a look at a normal plant cell, we know that it consists of a cell wall, a cell membrane, a nucleus, a large permanent vacuole, and other organelles like chloroplast, mitochondria, and endoplasmic reticula. If we had to compare this to a xylem cell, the nucleus, vacuole, and all other organelles will not exist, including the cell membrane. This produces a dead cell, which helps by allowing the free passage of water. Now, these dead cells are then stacked upon each other to create a type of tube-like structure, kind of like a straw. The cell walls are then thickened with a substance called lignin to strengthen the tubes, and this helps to support the plant. The end walls in between these cells are then destroyed, which also helps with the continuous mass upward movement of water. Now remember, xylem can only transport water in one direction, and that is upwards, from the roots to the leaves. So here we can see a photomicrograph of the xylem vessels. So imagine you're looking from the top of the plant into the xylem vessels. Firstly, we can see that they're kind of tube straw-like structures with tiny little ridges on the inside. So these ridges show where the cross walls of the xylem cells have been removed. So they show all the individual cells packed on top of each other. Then here we can also see how the cell walls of the xylem vessels have been thickened by lignin. Now remember, this is going to help to keep the plant upright and give it structure. Unlike humans and other animals, plants don't have a skeleton or bones to keep them upright and to give them a form. So they use mainly the xylem vessels as well as cell walls to give them their structure and their form. Next, we're going to have a look at phloem. Now, phloem is responsible for the transport of food materials. This would be mainly sucrose and amino acids. These materials are made by the plant in the photosynthesizing tissues like leaves and then transported to non-photosynthesizing regions in the plant. Then they can be used in respiration or other metabolic processes. Unlike xylem vessels, phloem vessels are made up of two structures. The first structure, which is the continuous tube where the food materials will be transported in, is called the sieve tube element. It has a cell membrane, meaning unlike xylem vessels, it is a living cell and certain processes can take place inside of it. The cross walls in between the cells are also not completely destroyed. Instead, tiny little holes called fenestrations are produced in between them so that the food material will be able to be transported in more than one direction. So sucrose, for instance, can be transported upwards as well as downwards. This is because photosynthesizing tissues aren't only found in one part of the plant, but in many different parts of the plant. And non-photosynthesizing tissues are then found elsewhere on different parts of the plant. The second part of the phloem vessel is the companion cell. Now, the companion cell will have all the little organelles as well as the nucleus. Between the companion cells and the sieve tube element, there's also tiny little holes, which facilitates movement of ions and different materials between the two cells. The main function of the companion cell is to keep the contents of the sieve tube cell alive. Because the sieve tube cell only contains cytoplasm and no nucleus or mitochondria, it needs the nucleus and the mitochondria from the companion cell to help it stay healthy. The companion cell is then also responsible for the uploading and the unloading of sucrose into the sieve tube element. So it's not just drawn up like water, but rather the companion cell takes up the sucrose or the amino acids and then puts them into the sieve tube element to be transported. Let's have a look at this photomicrograph of phloem tissue. So these little cells on the sides of the bigger cells is the companion cells, and these are the cells that would help to transport sucrose from the surrounding tissue into the sieve tube element. We can identify them as we can see that they're not hollow. They have a little bit of color to them. Now this color is mainly given by a stain used, but this stain will only color living organelles or living tissue. So we can see that it will contain cytoplasm and organelles and of course our nucleus as well. Now this bigger empty white cells next to it would be our sieve tube element. They are hollow and we can see straight through them. Now phloem as well as xylem tissue are found close to one another in structures called a vascular bundle. Now here we have an example of a vascular bundle as a photomicrograph of a dissection of a leaf. So if you imagine holding up a leaf petal and looking into the thin side of the leaf while it's cut open, if you had microscopic eye vision, you'll be able to see the structure. We use a microscope. So the small cells at the top would represent the phloem tissue that's responsible for the transport of sucrose. And the large cells at the bottom would represent our xylem tissue that's responsible for the transport of water. Here we can see the big difference in size between the phloem tissue and the xylem tissue. When studying to remember where to find the xylem and phloem tissue in a leaf, you shouldn't try and remember phloem being on the top and xylem being on the bottom, because you do not know in which orientation they'll give you the leaf. It's better to study it as the different sizes in the different cells. Next, we have a dissection through the stem of a dicotyledon. So here the vascular bundles are pretty observant, all of them in a circular pattern around the stem. Now, if we look here, on the outside, we can see the phloem tissue. Those are the small cells. And then on the inside of the vascular bundle is our xylem tissue. Now here you can use the fact that phloem is always on the outside and xylem on the inside to remember the position of the two transport tissues. One way that I remember which one's on the outside and which one is on the inside is we get tiny little insects called apids. Now they like to drink a sugary solution from the stems of plants. Now their mouth parts aren't very long, so obviously the sugary solution that will be found in the phloem should be closer to the stem or the outside of the stem of the plant. Next, we have a dissection through the monocotyledon stem of a plant. Now remember the difference between dicots and monocots is that a monocotyledon obviously has one cotyledon in their seed, a dicotyledon has two. So if you think about a dicotyledon, think about trees, and if you think about monocotyledon, think about grass. Now here we can see the vascular bundles are much smaller than those of the dicotyledon. They're also not in a regular pattern. They're kind of spaced out and positioned randomly across the stem. They're also orientated in different ways. So we cannot remember that xylem is on the inside and phloem is on the outside. We'll have to have a closer look at the stem or the vascular bundles themselves. So if we zoom in and we have a look, we can see that the smaller cells are the phloem tissue, and then the larger cells would be the xylem tissue. Okay, so now we're going to have a look at photomicrographs of roots. And the first one we're going to have a look at is the dissection from a dicotyledous root. Now we can see that most of the root is made up of these cells, and they're called cortex cells. Then at the center of the root, here by the pink part, that would be our vascular bundles. So the little X made by big cells would be our xylem tissue, and I actually remember it as the X for xylem. And then the little small cells in between the arms and legs of the X, if you want to see it that way, would be the phloem tissue. And now we can have a look at the monocotyledon root. So here we can see the dissection, and the one nice thing about this dissection, we can also see tiny little root hairs on the ends. We can see most of the cortex cells as well as the vascular bundles in the center. Now again, the large cells would be xylem, and then the small cells that are accompanying it on the more of the outside, that would be phloem. The last type of cell that we're going to have a look at today is the root hair cells, and these are individual cells on the edge of a root and is responsible for the absorption of water and mineral ions. Now, if you can remember from our previous chapter, water is absorbed through osmosis and mineral ions through active transport. Let's revise on how a typical plant cell looks. We know that it has a cell wall, a semi-permeable cell membrane, a nucleus, a big permanent vacuole, and then of course mitochondria and chloroplasts as well. Now if we had to compare that with a root hair cell, the root hair cell also has a cell wall with a cell membrane, but the surface area of the cell wall and the cell membrane is increased by producing a protrusion from the cell. It still has its nucleus and a very large permanent vacuole, but it doesn't have any chloroplast. Rather, it has a lot of mitochondria. This is because the root hair cell will be found on roots that are underground, meaning that there's no sunlight for the chloroplast to be able to photosynthesize. They would need a lot of mitochondria though to be able to transport ions through active transport. Remember, active transports require energy, and mitochondria is responsible for producing energy through aerobic respiration. Now, the increased surface area also helps with the absorption of water. Because it has an increased surface area, it means more water would be able to move through osmosis from the soil particles into the root hair cell. So here we have a photomicrograph of a root hair cell, and we can see the nucleus as well as the cell wall and the cytoplasm inside. We can see the protrusion of the root hair cell as well, which would increase the surface area for the absorption of water and of course mineral ions through active transport. Let's do a quick summary between the differences of xylem and phloem. Now remember, xylem is dead cells, so they do not have any living organelles present inside of them. So they lack cytoplasm, the cell membrane, and all the other organelles. Whereas phloem is living tissue. They have cytoplasm, and they also have companion cells that contain mitochondria, a nucleus, and other organelles to service the sieve tube element. Phloem tissue also transports carbohydrates in the form of sucrose in multiple directions, whereas xylem only transports water and dissolved minerals in one direction, namely upwards. Xylem also have cell walls that are lignified and has no end or cross walls in between the cells, whereas phloem has cross walls in between the cells, but these cross walls are perforated for the multidirectional movement.